Good afternoon. My name is David Cassak from InVivo, and welcome to the second in the series from the Innovators Workbench. Imagine waking up every morning and realizing that you invented coronary stents. That's really the warm, fuzzy feeling that our guest tonight, Julio Palmaz, has. Commercially, clinically, it's hard to think of another medical device over the course of the last 10 or 15 years that has had the impact of a coronary stent, reaching 80% penetration within just a couple of years and creating $1 billion market. Julio, in his modesty, will tell you it was a matter of serendipity, but it was as much a matter of perseverance. And in fact, despite how unique coronary stents are, in fact, a lot of the challenges that Julio ran into along the way are very reminiscent of challenges that other medical device entrepreneurs and inventors run into. So here to talk to us about coronary stents and what it was like to develop them is Julio Palmaz. Julio, I know you grew up in Argentina. Talk to us about how and what brought you to the United States.
Well, essentially, reading the literature in the early years about what the leaders were doing at that time is that I had an understanding what a successful academician and successful academic pursuits were, and started dreaming about it. And pretty soon I realized that I was doing medical practice, which was satisfying, and teaching, but research was certainly out of question in Argentina, and started looking north. And first I contacted some of those leaders that I admire through their work, and I was lucky enough to get positive feedback and eventually met some of them, and Stuart Reuter in particular became my lifetime mentor.
Mm-hmm. I know that the idea for a stent first occurred to you is seeing an early lecture by Andreas Günzig. Talk to us a little about when that was and how the idea for the stent actually came to you following that lecture.
It was actually second year of my residency training in the States. I went to one of the early meetings of the Society of Cardiovascular Interventional Radiology in New Orleans, 1978. And the keynote speaker was this young professor from Germany, Andreas Günzig, who was coming to the States to report on his early experience with balloon angioplasty. So I was very lucky in being in the audience, of about much smaller than this one here. And Günzig was a very charismatic and incredibly intelligent individual, explained the early experience about balloon angioplasty so clearly and in such a lucid description of the benefits and potential risks that when he came out to the reasons for failure, elicited in my mind, it could have elicited in anybody else's mind, I believe, the potential of a solution. And
what were those early failures? I mean, what did you hear Günzig say that told you that balloon angioplasty alone wasn't going to work, that something like a stent was needed?
He had a very graphic way to describe it. Said balloon angioplasty works sometimes and sometimes it doesn't. He said you would hope that most of the times it will be like putting your foot on snow. It'll depress the lesion and the results will be permanent. But many times the plaques behave more like rubber. You will apply the balloon and then the lesion will spring back. And sometimes the artery wall will delaminate and flake off, and things will end up worse than at the beginning, and you will have a complication. And he showed X-rays and specimens of microscopic specimens of vessels that had undergone angioplasty and had a complication, and that was essentially the prime for me to think about the solution.
Now, the stent was your first and really your major medical device development. You hit a home run right out of the box, but were you a tinker? Are you good with your hands? I mean, did you pull watches apart as a kid and put them back together again? What took you from the leap of, hey, there's something else needed here too, I'm going to develop this product?
I was a tinkerer, yeah. As a kid, I grew up, playing with things and doing simple things. So I was a tinkerer, yeah. And I thought mechanically too. So faced with the possibility of a vessel recoiling back and delaminating and things like that, it was not difficult for me to come up with the idea, why not to put a cage there that will hold all that material back and keep the vessel open.
So talk about your early efforts to develop the stent, because as I understand it, began with a trip to Radio Shack.
Yeah, actually began more like by writing down my idea on a piece of paper. In fact, on the way to the airport from that meeting, my chairman, who was always lecturing me about what's the way to do the things better, I told him about this early idea, and he said, write it up.
Just write a monograph on this.
Right. He said, no, it won't be a paper, of course. He said, but it'll give you the habit of putting down in writing what is in your mind.
Mm-hmm. You said somewhere that document, which you never published, became an important document later on. Why did it become important?
It sure did. It was one of the lucky strikes. I wrote my idea down. Of course, I looked a little bit into the bibliography and saw that, come up with some intelligent statement of the problem and its potential solution. Distributed among my superiors at the time, my faculty members, and one of them returned the monograph to me with a note and a date, and that became an important legal document because it became a date of conception.
Was it your basis for intellectual property?
Yes.
So take us back to Radio Shack, because you suddenly thought that there was this device that you could develop, but you needed to find the materials for it.
Right. Well, I mean, the idea was, it evolves slowly in your mind. The idea of putting a cage that would be driven by a balloon, which was the original idea of Grundstig. The Grundstig balloon became available months after he visited us here in the States. And I had the opportunity to do a few angioplasties with my faculty members, and I experienced the complications in my own hands. So we did have the failures that Grundstig described, and that was a very important reinforcement to pursue the idea. And also, besides writing the monograph, I tried to put a little prototype together. And I started playing with, like you said, I went to Radio Shack and got myself some winding copper wire and some solder materials and solder gun and so forth. And went home and made my first mandrel out of a pencil, sticking pins on a pencil about an inch apart, and then weaving with copper a mesh around the pins. And then once that was done, I would pull the pins apart and the thing will not hold together. And you would put in a balloon, but it wouldn't have any plasticity to it. Then obviously the solution was to fix the cross points, and that's where I started using the solder. Mm-hmm. And then once that happened, yes, the cage did have plasticity. It could be collapsed onto a folded balloon and then expanded again by inflating the balloon. Mm-hmm.
Now, I understand there's a story that the idea for the cage design actually came from a carpenter was working at your house and he'd left some materials around. What's that story?
That was later on. Once I was confronted with the need of using two materials. Of course, I was never going to use copper. Mm-hmm. And by reading a little bit about dental implants and others, the type of prosthetic implantation materials, I realized that it probably would be some sort of a steel. But then if I needed to put the cage together with some sort of a solder material, then silver was my only option, that at least I knew at the time, because that's the way guy wires were put together. And that apparently was, according to my limited understanding of implant materials, that it was a no-no, that you had to use materials of single construction materials. It would be the best scenario. So the solution came one day in my garage where I had my workbench and used to tinker with things. I saw on the corner of the garage a piece of the metal lath that the mason was using to make an expansion of the garage. And I don't know if you're familiar with the material. It's essentially a crisscross mesh that is nailed onto wood, and then you apply cement over it and hardens up and so forth. Well, this material was essentially crisscross mesh, and by playing with it, I first put it on a vise, collapsed it down, realized that the diamonds were actually stagger slots when it was in a collapsed state. And obviously right there, I said, well, if you start out with stagger slots, it can be made on a tube, then I don't need to use any kind of material to put it together. Then the single piece material construction was, was essentially suggested from that material.
How are you getting feedback on what would work and wouldn't work? You were years away even at this point from doing anything kind of like animal studies. How did you know that certain materials were going to work and others weren't, and certain designs were going to work and others weren't?
Well, for one thing, I used rubber tubes to mimic vessels, and I would put rubber tubes nailed onto a board, and then try to navigate, the lumen of the tube with a catheter and guide wires, just the same way you do it with arteries, and then blow the balloon up there, and noticing that that the stent actually would deform the tube by expanding it at the site. Then you deflate the balloon, pull it out. At the beginning, obviously, I was yanking the thing out, but then I kept improving it until finally, I was able to implant this remotely. And but the rest was essentially an extrapolation for what we used to do at the time. Balloon angioplasty was already going in the late 70s.
So your first implant was in what? Was it a rabbit or a pig in around what, April of 1984?
Actually, the first animal implantations came when I moved to Texas. So, but by that point, I already had gone quite a ways into my prototyping work, to the point that I decided to move to Texas to carry on my my idea of doing this into a serious research project and do the animal experimentation and so forth. That would require protocols that need to be approved by animal research facilities and so forth.
Well, let's get you to Texas, but let's be back here because you were working over at Martinez VA Hospital here. What were you doing there? Basically research on this project?
No. All the research, I didn't have research time allocated in my activities. It was all clinical. The only work I did at home of hours. And as I said, I need to move to Texas to be able to do my first animals. I did not have the opportunity to do implantations or any degree of important development in California.
Several people I know played key roles in the eventual development of the stent. One very early on, one obviously was Richard Schatz, and we'll talk about him in just a minute. Another was Philip Romano, and we'll talk about him in just a minute. But one was Werner Schultz. Who was Werner Schultz, and how did you meet him, and what did he help you solve?
Let me step back a little bit. The motivation for me to talk to companies was the fact that I could do large prototypes. In fact, most of the geometry about stents and expandability ratios, foreshortening, open area, and so forth, width and of the material and the slots and slot ratios and so forth, I did it with cardboard models. The cardboard models were, for me, far more useful than anything else. I could make them large, and I could play with, measure things and so forth. The big challenge was miniaturization. I knew that the minute I needed to get into coronary sizes, I was talking about things I could barely see with your naked eye, that any hand technique, or hand construction, it was way out of the question. That will have to require some sophisticated technology, which I knew nothing about. So at that point is that I decided that a company needed to get involved, and I actually talked to two companies, while I was a resident in radiology. One was Hancock, then became Vascular, and the other one was Shiley Corporation. Both of them looked at the project for a while, got initially interested and eventually turned it down.
Why?
Well, actually their answers were pretty similar. There was the general distaste for metal implantation in arteries. They foresaw the advantages. They recognized that the stent would have a mechanical advantage in solving the problem of dissection and recoil, but they thought that metals did not have a future as vascular implant. In fact, was part of a prevailing thought at the time. There were so many other techniques that were being evaluated at the time that stents had to wait their turn. You know, it was laser and it was atherectomy, and even balloon angioplasty was not totally proven. So all these techniques needed to go through their paces and before stents would actually have a consideration.
So one of the product companies, was it Shiley or was it Hancock, told you about Warner Schultz?
No, it was actually the Cook company. When those two companies turned me down, I contacted the Cook company. One of their people working in R&D told me, no, we are not interested in implantable metal either, but we can recommend you a person that might help you. And they gave me the phone number of Mr. Schultz, who was actually a former retired technician of Stanford University. And Werner was living in Willits, California, and in retirement with his wife, and gave me a call. He was very helpful and very kind, invited me to go visit him, and I did.
And what did he tell you?
Well, I took a 3-hour trip over there with my family. Werner was living in this log cabin in the middle of the woods, very pastoral environment, and he took me to his garage, and to my surprise, he had all kinds of high technology equipment there. He was making needles for micromanipulation of cells and things like that, and I was totally surprised. I mean, you wouldn't expect that in that place. And so I took all my prototypes and my drawings, and over a table and tea and rhubarb pie, I explained what I was trying to do, and he said, well, I told him about my motivation to walk away from the woven design because of the need of solder. And he said, well, you don't need to use solder, you can use arc welding. And that way you don't need to introduce another material. And right there, he started making drawings about how to make an arc welder for small wires. And then I said, yeah, but this other design is more interesting, I told him, because I could start, with a solid tube and put these staggered slots. And he said, oh, there are at least 6 or 8 different ways to make this. I said, well, I'm anxious to hear the first one. So, I was taking notes as fast as I could, and he mentioned EDM, water jet blasting, ion bombardment, electrochemical etching, mechanical stamping, and he went on and on and on. And he said, but start with EDM. He said, EDM is your technique.
EDM stands for?
Electromechanical discharge machining, which I knew nothing about, of course. It was the first time I heard about it. He said, there are lots of shops in the Bay Area that do this, and they're very good to do miniature devices. I would recommend that you contact one of them, and you'll get your prototypes from them.
But Warner never saw the first stent developed.
No, in fact, I was fascinated with the man, and I wanted to start a relationship, and I exchanged a couple phone calls and a letter, and one day I called back, and they told me that he was dying in a hospital, and so Werner passed.
If I'm right, it's around this time that you moved to Texas?
Yeah, that to me was that, and my chairman, Stuart Reuter, who had already moved to Texas 3 years before, was trying to recruit me there. and Schultz's death were pretty much the last drop. I mean, that was what I needed to decide. Okay, I had to make the move and go to Texas, and I did.
Was it in Texas that you ran into Richard Schatz?
Yes.
Now, his name's on the stent.
Yes.
So he obviously had some role in this.
Oh, he did have a big role, yes. About a year after I was in San Antonio, where we had an EDM machining campus, so I was able to very quickly get on the development program. I met Richard Schatz, but at that time, the reason why I met him was because I was looking for research funds.
So, tell that story. Is that where Philip Romano comes in?
Yep.
Okay, who's Philip Romano?
Philip is is an entrepreneur in restaurants, restaurant developer, and developer of other concepts, essentially a keen businessman and always looking for opportunities for business.
His restaurant chain was?
Well, he was the founder of Fuddruckers and Macaroni and Grill, and he had many other ones.
So maybe this was like a package deal. If you 10th cheeseburger, you get a coronary stent because you're going to need one.
That's right.
What interested him in this coronary stent idea?
Well, as I mentioned, the story starts with Richard. I was at the Southwest Research Foundation asking for support because already the Department of Radiology helped me with initial funds, but then eventually they loaned me money, and I had already animals with implants that I was not able to support anymore. So I was pretty bad need of getting a research fund. At the Southwest Research Foundation, Dr. McGill, the director, said, well, I mean, we cannot help you. I mean, we are not in the business of giving money. We actually need you to bring the money here and, get yourself some sort of a grant, and then you can do the work here. But by the way, there is another loser like you, he said, that is working with baboons and with an atherosclerotic model. And his name is Richard Schatz, and he gave me his phone number. So I, with a little hope of anything, I gave Richard a call. And Richard, when he saw the idea, he loved it. He thought he was very, very enthusiastic about it.
You formed a company at that time around the coronary stent, yes?
Well, about a year later, a few months later. But initially, we did a little bit of work together with Richard in my lab. And one day he called me and he said, I found a person that might be interested in investing in the project. And so he put me together with Phil Romano, and eventually Phil formed a business around this.
Who was advising you at this point? I mean, what kind of deal did you strike with Phil Romano and Richard Schatz? And how did you know how to protect your own rights and what was fair for an investor? Because I think, what did Phil put in, $250,000?
Yes. The thing came on the background of the university actually turned me down from my patent application, because it was a few months before that I actually had two major setbacks. One is I asked the University of Texas to patent this device in my behalf, and the patent committee saw no value in it and turned me down. And then the federal government, I applied for a grant to develop the coronary stent as a coronary stent project, and they also turned me down. So at that point I went to Southwest, and then I found Richard and Phil. But essentially, Richard was very enthusiastic about that, and he advised Philip on the potential value of this. But what we had at that point was so little in terms of animal evidence of feasibility that it really required somebody with big risk-taking potential, and
that was Phil.
And that was Phil. I presented this to Phil, I remember one morning in the library of our radiology department, and Phil came in with his lawyer, and he was sitting there in his three-piece suit and looking very impressive, and I was very nervous because I never talked to a venture capitalist before, and I was making all kinds of excuses about how preliminary this work was, and how little was any possibility of that becoming a success. So I said, this is a good way for you to lose your money, and it was a good project for me to write a paper, but I cannot assure you that this is, there's 100 to 1 chance that it was ever going to become a success. And so Philip sat there very quiet, and at the end of the meeting, and all my excuses and comments from the lawyer and Richard, which were on the opposite sides, Philip said, I think I can put this in a box and sell it for a lot of money.
Richard Schatz said this.
No, Philip Romano.
Philip Romano said this.
And so the more I insisted that this was risky, the more interested Philip became. And so pretty soon...
I don't think you were talking to real venture capitalists at that point, my sense is, but it's a different issue.
They eventually, Philip wanted to form some sort of a commercial entity around this. I wanted to stay out of it. In fact, I wanted Philip to do business directly with the University of Texas, but said, Philip, I don't do business with the government, I do business with you. And he insisted that we form a partnership. We eventually made the university part of this too. Oh. And so it became a very, very good group because I did most of the science side of work, and Richard eventually became the clinical, the principal investigator on the clinical trials for the coronary arteries, and Philip was the businessman. So we were a very effective group.
I know that until Philip got involved, you were financing much of this on your own and really basically depleted your funds. Eventually somebody, a lot of companies, made a whole schoonch load of money off coronary stents. At what point did you realize the tremendous commercial potential here, and how important, at what point was it important to you that you actually make some money off this device?
When Philip put about $250,000 in my research account at the university, we were able to hire 2 technicians, and I was already on the winning side because, as a researcher, I had what I wanted, which was my project was on the way, and I had a whole line of research already all stringed up, and I was having a lot of fun. I actually abandoned some of my clinical activities. I only took call at night, but I would work on research purely now that I had the money and the technicians and my lab well equipped. But in about a few months, it was almost close to a year, Philip supported my patent application, and we had a patent application, and we have enough evidence at that point that we could go to companies and tell them seriously that this was a very promising project.
So before we get to other companies, the other companies that you called on, from the time that you hooked up with Phil and Richard Schatz to the time that you were ready to take this prototype to other companies, what was involved? What did you need to do? What stage was the prototype stent, and what did you need to do in the intervening period?
At that point, it was just do the due diligence for the project was to do the research. There was a lot of feasibility and need to be proven. Histology that needed to be, essentially on the early animals, we noticed that there was something that was fundamentally right about the stent project, is that we put this device in there, and at that time we knew nothing about what the healing modes were going to be, but from the very beginning it was looking right. I mean, the stent would first be covered with clot, and then that clot will pretty much become translucent tissue, and the vessel remained patent. So it was doing the right thing. And then we found out that the tissue that formed on top of that cage was actually healthy vascular tissue with the normal lining of a vessel. So all that was, unexpected, and it was a very happy event. unlike the type of events that you typically find in research where 10 things go wrong for each one that goes right, with the stent, it was all the way around. It was everything we tried was successful.
But you did run into one major problem. Wasn't there early evidence of subacute thrombosis and thrombotic closure? Was that not at that point?
No, that was not at that point. No, that came later on in the clinical trials. But the stent, I mean, we tried it in arteries and it worked. We tried in veins and it worked. Then we tried it in conduits that we created, let's say across the liver parenchyma, and it also got covered with the same tissue. So it seemed that there was no end to the possibilities that this technology could offer. And we became very, very enthusiastic. And we had a very, very good body of scientific evidence that anybody could see at that point.
And this is about what time frame? 84, 85?
It was 85, 86. And we started actually showing, with Philip and Richard, just about every company that we could, we knew of. We showed the project and we offered them the possibility of taking this project as sponsors.
Who did you talk to? Because now this is your second go around and talking to product companies. Who did you talk to this time around, and what was the response again here?
We talked to everybody. All the companies that were involved in the catheter business, all the companies that were involved in the surgical prosthetics, valves. Of course, I didn't personally, but Richard and Phil contacted just about everybody.
And got turned down by virtually all of them.
The companies at that point were interested because the material we had was compelling, but not to the point to commit an important resource to it. So pretty much was the same story. Yeah, we would like, but if it requires a financial, involvement, we'd rather not.
I know that you had, this really compelling clinical data behind you, but did you get discouraged at any point? Did you ever think at some point, well, we've got really interesting data, but this is never going to turn into a product?
But not me, because, I was still having fun with my research and things were going well. I was going to the meetings and the findings of our research were causing a lot of attention, at least in academics. So I was getting my money out in terms of time and effort.
Now, the company that actually did take a chance on the stent was Johnson & Johnson. No surprise that they're sponsoring the meeting tonight, and they had tremendous success with in their cordis division. But it didn't start in Johnson & Johnson cordis. There was no cordis at the time at Johnson & Johnson. There wasn't even interventional systems. As I understand, it was originally put into the Ethicon business. How did the Johnson & Johnson connection come about, and talk about Johnson & Johnson's role in helping to develop this product?
That was actually Richard's find again. Richard Schatz went to an RSNA, I believe, in 1985 or 86, and he met Robert Ulrich, who was at the time a person working for Johnson & Johnson and who had the mandate to look for a way that Johnson & Johnson could get into the vascular business. They were a little bit, I think they had bought at that time an ultrasound company that was not a good experiment for them, but they were interested in the catheterization business. So they needed an interesting project to be able to start there. And the stent was perfect for them. And not that they didn't embrace it right away, but they looked at it with great interest, and we started talking.
And in fact, as I understand it, because they didn't have much experience in vascular medicine, they hired a consultant to kind of vet the idea. Talk about that. That's an interesting story. Talk about what happened when they put this focus group together to talk about bare metal stents.
We started actually, visiting each other. Representatives from Johnson & Johnson would come to San Antonio. We would go to Newark and so forth. But eventually, when they got serious about it, they hired a consulting company that would look into the feasibility of a prestigious company like Johnson & Johnson getting into a rather risky business like stenting, and if that would be a reasonable thing to do. So I believe as part of the study that this company did is they formed a panel of about, I believe, 20 or 30 consultants from all different walks of life. And they put him in a hotel room over a day. They presented them with my work. I didn't present it, but somebody else did. And then they started a debate, and I had the tapes, which were very interesting, about the comments. But the panel was pretty consistent. They all recommended against getting into the coronary stent business.
So what happened? Why did Johnson & Johnson do it?
Well, I mean, Johnson & Johnson, the people that had the decision power at the time, they still were considering stenting. They were, pretty drawn into it. They liked the idea. Eventually, the company group chairman, Mr. Larson, invited us to go visit with him in New Jersey. And over lunch, he asked us very simple questions, and he made the decision right there. Mm-hmm. Single-handedly, we will get into the stent business.
You described for me in the past that your relationship with Johnson & Johnson was a very good one from your perspective. How much work beyond what you and Richard had done did Johnson & Johnson have to do to make the stent approvable and ready for market?
Well, Johnson & Johnson took a lot of risk. They did take a lot of risk. For one thing, the financial commitment that they took at the beginning was important. Mm-hmm. They formed a company. At the beginning, the responsibility for the stent development was given to Ethicon. And Ethicon, being in the suture business, they thought that it was the right company for it. But then they decided that they were going to form a company with a special interest, and they formed Johnson & Johnson Interventional Systems. Right. And at the beginning was a group of 10 or 12 people, and then it grew quickly. But they did take enormous amount of risk, and because obviously they were deeply into the stent business. At the time, stents were still controversial. There were big editorials coming out in the papers, and against stents. Remember the dark days of the the early trials of coronary stenting with very high rates of subacute thrombosis rate. The editorials that came out in the specialized press and in the public press, and talking against stents. And so, Johnson & Johnson was really put through a lot of trials. Yeah,
and not surprisingly, the regulatory path was particularly difficult. I know you got your first application for Illiac was approved, but your first application for coronary stents was turned down, if I'm right. Yeah.
That's
Talk
right.
about that.
The Illiac stent trial, who was the principal investigator for that trial. It was fascinating to me. I'd never been PI on a project like that, and they gave me the opportunity to go around the world and training people and gathering cases, monitoring, and helping the clinical specialists gather data and monitor the trials and so forth. Then eventually I went with the Johnson & Johnson people to the FDA, and I had the opportunity to defend the project myself in front of a panel, and we passed. And the stent got approved in 1991 for iliac artery use. And so that was a very happy, well-rounded experience for me. The coronary didn't go that way, and it was a little bit more tortuous. The initial trial had some difficulties. Of course, we, like you mentioned before, we encountered these problems of subacute stent thrombosis, and we had to do some, work around, anticoagulation regimens and the risks involved in chronic anticoagulation and so forth. But eventually, on the first presentation of this prospective non-randomized trial presented at the FDA, for the first time, the panel rejected at the panel session the project. And Eric Topol was the chairman at that time, and he had his good reasons to make an argument for rejection, and he prevailed, and the first trial was rejected. It was a big blow for the company and for us.
Did Johnson & Johnson ever think of abandoning the project at that point?
I believe it was. I think there was a motion from the headquarters to actually pull the plug on the project and abandon the coronary program.
And why didn't they?
Well, I think they didn't. Somebody in the group made a good case to go forward, to endure the storm and undertake this big enterprise, which was to do 2 trials of randomization trials, one in the States and one in Europe, the stress and stent trials. And they had to be done in a very short period of time at a very high cost, and it would require an enormous amount of coordination and effort, and it was done.
Mm-hmm. Yeah, in fact, it was interesting at that time, notwithstanding the fact that Eric Topol headed the panel that rejected you, he was one of a number of leading interventional cardiologists who were enthusiastic about stents from the very beginning. Marty Leone, I know, was an early supporter. Eric Topol was an early supporter. Was it hard or easy to round up interventional cardiologists to do these trials?
Eric Topol was the first person that invited me to give a talk on the stents, and invited me to go to Ann Arbor, University of Michigan, and I presented the stent project to his group. And he was very enthusiastic about his stents. Mm-hmm. But he had a responsibility as chairman of the panel, and he thought there was not enough evidence, that Johnson & Johnson had not done their homework, and he made a case for rejection. Mm-hmm. And he was probably right.
So the stent was finally approved in April in 1994, and within 4 years, this I think I've been told is the record for the quick adoption of medical devices. Stents were used in 80% of percutaneous coronary interventions. Did the pace of adoption surprise you?
Well, I mean, there was already a sample of how things were coming in the future, because by the time that we got the disapproval by the panel, the FDA panel here in the States, the stent was being used in rapidly increasing numbers in Europe and in Japan. There were some investigators in Japan, like Nobuyoshi, and Erbel in Germany, and so many others around the world that were using the stent very successfully. Antonio Colombo came up with the idea of using high pressures, and I started putting aside the oral anticoagulation in favor of aspirin, and so forth. And so things were starting to look good in Europe at the time that we had these setbacks here in the States. So that was probably the reason why there was enough support to go on. Mm-hmm. But like you said, there was never an idea of how much the stent was going to be used. I mean, I thought at the beginning, every month that we change our numbers, we thought perhaps the stent was going to be used in 5% of the cases, maybe 10, 20%, but it was like, as the years went by, we were always surprised by the adoption of a stent in such an increasing numbers.
What was the rationale? It's very interesting because, again, 80% penetration has been so successful. When you thought about it being used in 5 or 10 or even 20%, what was the rationale for a niche use? Why did you think that it might not? Why did you think that what was the argument against that it would be widely adopted?
Well, because of the failures at the beginning. At the beginning we had subacute thrombosis rates was as high as 5%, and that was a dreadful complication. Mm-hmm. And we didn't know that we were going to be able to overcome that. Mm-hmm. When we started using potent oral antiplatelet agents and stopped using Coumadin, things got all of a sudden much better. Mm-hmm. And the stents really became like a good, safe tool. And the investigators were starting to use the stent. You see, that is an important issue. The early stent, the Palmaz Schatz stents, were difficult to use, and required the good investigators, the people with a lot of skill, were able to put it anywhere. But the average guy, the one that was not trained, had difficulties with it.
Were you aware of that? I mean, because that raised an interesting question, because, I mean, it took you nearly a decade to develop the first stent. Johnson & Johnson had a monopoly for a couple of years, but really within 2 or 3 years, they were seeing the next generation of stents come along from Guidant and AV. Did the speed with which the next generation came to market surprise you at all?
Yes. Yes. It did surprise me.
Did you just not anticipate that there'd be a next generation, or did
No, in fact, even when we agreed on using the Palmaz Schatz stent as the first stent, we neither Richard or I liked it. I mean, we thought it was a clunker. And, but we were limited at the time, not by the design, which, you know, at the time we already were thinking of other ways to connect stents together and to make them flexible, but rather by the manufacturing technique. The only thing that was available at that time for industrial use was EDM, electromechanical discharge. And the way the electromechanical discharge works is by making straight cuts. So that's why the design of a stent had to be essentially adapted to that type of manufacturing. Laser became available 2-3 years later. And when the cool laser came out, of course, you know, the free-forming and cutting of stents opened the possibility for all kinds of different designs. But, you know, before the PS stent was approved internally as being the first product, we already had all kinds of ideas about how to make other designs.
Did you have a continuing relationship with Johnson & Johnson? Did you have a was your relationship with Johnson & Johnson ongoing at this time, or had you? Oh,
yes. No, yeah, when I mean that we were talking about all these things, you know, it was already as part of the developing group of Johnson & Johnson and us.
I know Johnson & Johnson took a lot of criticism for allowing themselves to be past, technologically speaking. Were you urging them to do, I mean, what's your view on, they lost 90% of their market within the space of a couple of months. What's your take on what happened in the next generation of stents to Johnson & Johnson?
Johnson & Johnson is an incredibly ethical company, but it's a very large company. And they move relatively slowly, just because of that size. And I think that's the reason why, probably why Johnson & Johnson and us lost the opportunity back in '96. Also, they took over Corys Corporation at the time, and that might have been a distracting factor. But I really cannot speculate. The reason is that we did lose the opportunity.
You mentioned that you had already thought about some future designs, and we've got just a couple minutes left. Let me ask you about 2 future areas of stent design, because stents have been tremendously successful, but I think everybody knows that there's future horizons beyond this. At last year's TCT, you presented a session on nanotechnology and stents, and I know you've been working with a small startup called Advanced Stent Technology. Where do you think stent technology is going from here?
Well, you know, stents have become a revolution, as you said. And the thing grew on, you know, despite of all our efforts on the side. I think that the most incredible thing about stents is how they help developing the catheterization discipline. There is thousands, tens of thousands of cardiac catheterization laboratories around the country, tens of thousands of people that can man them very well, and there is an enormous industry behind this. Physicians can put a device now anywhere they want in any organ within minutes and incredibly safely. So I think the stents have a tremendous opportunity, whether they're called stents or something else, to become a platform for interactive technology. I don't see why stents cannot be used in the future to interrogate problems in organs using nanotechnology approaches.
What about drug-eluting stents, which are obviously will create another market of magnitudes greater than bare metal stents, which was significant. How wide did you think they'll be used, and will they make bare metal stents obsolete?
Well, drug-eluting stents have had a tremendous success. I mean, the benefit to risk ratio they have is very large, and they do the work. And today they offer a definite advantage over bare metal stents, particularly in certain patient subsets. So it'll be no point to actually criticize that technology. Nonetheless, in my view, I think I can make the case for a technology that was developed without understanding what we're trying to fix. Because in reality, we never understood restenosis very well. And I'm not sure that we did our best effort to try to understand it. I mean, at the beginning, we had some rationalization about that perhaps it was design that caused restenosis, and then we justify the creation of new designs based on that. But when all that is said, you see all the stents today are very similar. All the second generation stents are almost identical. I mean, they have very small differences among them, and they all work equally well. Nonetheless, there's still restenosis. So something else has to be there, and now we have to think of materials. So I think the case for material-induced restenosis has not been made very clear. And I think that we are using a very powerful suppressant technique without really understanding what we're suppressing. The other thing is, and this is not my opinion, it's many others, is that as years go by and the restenosis rate on drug-eluting stents creeps up, the difference between the best performing bare metal stents and the late restenosis rate on drug-eluting stents is starting to get so narrow that the justification for widespread application is starting to be lost. So at least my opinion, and it's strictly my opinion, is that drug-eluting stents may be actually useful in certain patient subsets in the future, but not for widespread application.
We think about bare metal stents and drug-eluting stents as being so clinically compelling. Do you think cost will be a factor at all in deciding whether because I've heard the same arguments that you just put forward, that the success of bare metal stents compared with the next generation of studies on drug-eluting stents, we're not going to see the 0% restenosis rates that we saw in FIM and Ravel, will make the difference between the two much smaller. Will cost be a consideration for interventional cardiologists in deciding whether to use a bare metal stent versus a drug-eluting stent?
Well, the cost consideration is already an important one in Europe. As you know, the Europeans are not embracing drug-eluting stents widely, only based on cost considerations. And in the States, I think it'll be less of an impact. I think here we have the tendency to adopt the technology even if it is expensive. And reimbursement issues seem to be solved even before approval. So I think that will not be much of a problem in that respect.
Well, great. The story is just a fascinating story and extremely well told and a challenging one. I guess one final wrap-up question, and that is, having invented the stent, did you ever think of anything else?
No.
No? Okay, well, thank you very much.
Thank you.
I guess my question for you is, what advice would you give to somebody at our stage who may have an interesting idea and is just not sure how to get it going? Maybe a good way to answer that question is, if you had it all to do over again, what would you do differently? What advice could you give to some of us?
Yeah. Okay, definitely practical advice is every time you have an idea, write it down. Yeah. Because so many good ideas are just lost in that, and time goes by and all of a sudden you lost your track of time or you lost your chances of establishing your date of conception. Always write it down. And second, my advice is perseverance. I mean, you will have more reasons to drop a project than you will have to pursue it. So if you believe in the idea, just don't let anybody, no expert, to come and tell you that it won't work. If you like it, just keep on working on it.
A follow-up on that question. I take it back in the late 70s and early 80s when you were working on the stent design that you were alone in the field in doing this, that when you were conceptualizing this, there weren't a lot of other people working on stent designs. How much did you weigh back and forth, talking a lot about this, going to colleagues to get feedback, to talk to companies to get funding, talking to others to vet the idea, versus I better keep this to myself because no one else is really working on this and this can be something really special and I don't want other people latching on. So I know a lot of people, particularly in the device space, have this tension that if they talk too soon, too early about their device, they risk giving something away or tipping their hand to somebody.
That's part of a serendipity. I mean, of course I could have given this away a million times, but I was fortunate. The first deal with honorable people that, honored the confidentiality agreements we had, and second, that there was no interest. And that's probably the most important thing.
That's actually a lead into my question. I'm David Benarin. I'm a physician. Did my first invention and venture startup actually just out of high school a while ago. Couldn't do that now. So the question becomes this. Your story's interesting. You struggled to get your product out. You worked, you showed it to people, they said no. And having sat on both sides, at the successful and unsuccessful side, how do you decide, I'm talking to myself sitting on both sides, how do you know when you are a brilliant innovator, a future seer, who has to drive really hard in order to bring a product that needs to come out into the eyes of everyone? And when do you become the pig-headed inventor who is so enamored with his own technology that he can't realize that it's not relevant? And where's the balance there?
I think you give your own, you have the answer right there. Again, no, you don't, I didn't actually set out to make all those considerations. I just pursued it. It was a fun project for me. I mean, I cannot tell you that I had this, crusade that I had to go through with. It was fun. I was doing my academic project. It was allowing me to, because of its novelty, to go and compete for time and attention in the meetings with other people that really worked very hard for other things that they were presenting. For me, it was, a simple, crazy idea, and it took a life of its own. But I'm sorry, I didn't make a lot of considerations as I went along.
It's a follow your heart answer, it sounds like.
Pretty much something like that.
Thanks.
Hi, Sekou Crawford. I'm a design student in the Biodesign program. So you commented on drug-coated stents and talked about how they may not compare very differently to bare metal stents. We're researching a percutaneous solution for valves, and there's been 2 heart valves to date placed at the aortic position on stented valves. So I thought you might comment on stented valves, how you think that might play out, and other technologies that kind of build on stents as a basis for implantation.
The percutaneous valves is a fascinating project, but I think it's an order of magnitude more complex than stent grafts. I think the engineering problems surrounding those valves are very large. Perhaps as a bridge to surgery, you may convince the FDA that a valve, even if it survives 6 months, it may be justifiable as a trial in patients. But I think the mechanical requirements for a percutaneous valve are so high, to keep a low profile compatible with percutaneous introduction, it's going to be very, very hard. All that said, it's going to happen. Somebody is going to figure it out, and valves will be with us, I'm sure, sometime in the next 20 years. But I don't think the development is going to come very easy.
I'm a graduate student here in chemical engineering, and you mentioned basically that in the future you see sort of an integration between MEMS, nanotech, and medical devices. It seems that we're entering an area, and I mean, in many ways I think you led this, where traditional physician responsibilities are being changed because of other people with different ideas coming in from different areas. Like microelectronics is a good example, as you mentioned. I'm wondering if you can give any advice to folks in the audience who are not physicians, but who think that they might have interesting value to add to products along the way. How you work with physicians, how you gain credibility within the medical community if you don't have an MD, but you have an interesting idea that might be in an emerging area where traditionally there has not been that sort of a medical device. So maybe you can frame it in sort of in the framework of MEMS, let's say, if you have an interesting idea or you've talked to people in that area.
Thanks. That's a very good question. The problem in medicine is that there is typically no interest or no money for development of fundamental technologies. But the important thing is, at least in my viewpoint, to be aware of what's going out there. And whoever has the ability to see the opportunity between something that just developed in field A and to apply it to field B, then is the one that is going to reap the benefit. I think microelectronics is a good example. A lot is going on in microelectronics and nano devices in general. Typically those things have an enormous developmental cost, but very low production cost. If you can actually identify an area that of technology is mature enough to be applied to medicine, and if you are clever enough to figure out where it's needed, then the opportunity is incredible. And I would see a smart stent for an interactive information, say on kinetic activity of the heart, electrophysiology, even gas chemical information and so forth, being far more feasible than a percutaneous valve.
But let me just ask a follow-up question to that, because you're early, well, not early, it was relatively late, but fairly late into the process, you began to attract a lot of leading interventional cardiologists who got excited about the idea. Marty Leone, Eric Topol, the guys like that. How did those relationships come about? Do you think it was the idea that they got interested in the concept of the stent, or between you and Richard, were you able to open doors by virtue of being physicians that non-physicians might not be able to open? I
mean, those guys that you mentioned became leaders not because of serendipity. Those were leaders back then. Yeah. Marty Leon, when we first contacted him, he was already known to be a future bright star when he was working at the NIH, and he sure followed that path. Same with Eric Topol. I mean, but in many circumstances like that, it was them that sought us out. They identified an interesting area, and they came, and Eric Topol being a good example, being the first person that ever invited me to give a talk. And he was, I believe, an associate professor at the time. He was not in a leading position like he is today. So I think those individuals, they're very, very, very smart and very aware of what's going on, and they see these things coming.
Next question.
My question has to do with, you said there was a paradigm shift in the clinic once the next generation of pharmaceuticals came along, having to do with coagulation pathways, and what your thoughts are as far as antisense or other things that might improve dramatically the clinical outcomes from the pharmaceutical side combined with the device side.
That's a good question. I'm not sure I have the answer for that. I don't know if we have reached the limits of what we can do in terms of optimizing the healing patterns of a stent and early events like thrombogenicity and so forth.
Do you think, though, that you'll see a reduction in the use of heparin and more of these next generation compounds, or how do you see clinically what the optimal, if you had to pick an optimal drug regimen now, what would be an example of an optimal drug regimen in interventional cardiology, in the acute setting at least?
Obviously, people are working on antithrombogenic, the HIPAA code is one of them. But I'm not sure there's a big motivation to really develop more antithrombogenic coatings, because the antithrombogenic regimes today are so safe. So I don't think there is a big push towards decreasing the use of for example, Plavix and aspirin, even taken up to a year, is still very safe. So when the incidence of bleeding complications is so low, that I don't think there will be a big motivation in that area. Now, restenosis is a far more difficult problem.
Okay, thank you so much.
Sure.
Hi, my name is Ross Evans, and I'm a fellow with the Biodesign program. Something that's interesting to me is a little bit of your background, and that you come from Argentina. I haven't spoken too much about that, and I'm curious because I'm wondering if you have a perspective. I am particularly interested in trying to apply new technologies to developing world needs, and not that Argentina is particularly a developing world, but it's surrounded by it being in South America. And I'm curious if you have any particular insights about how we might cause these technologies to be more broadly dispersed throughout the world, as well as maybe insights into different problems that are seen in that part of the world or in the lesser developed world that if you've thought much about that in terms of trying to apply your knowledge to that specified area.
You know, in days, the gap is widening, you know. I mean, at that time, you know, in the early 80s, you know, a tinker had a chance. Today, things are far more difficult. You need to be part of a big supporting system in terms of technology to be successful. The possibility of low-tech contributions is much decreased compared to, you know, 20 years ago. You know, my exposure at that time, being a resident of Argentina, was pretty much the same I would have here, because I was reading the magazines at the same time that everybody else was receiving them. So if you're an avid reader, I mean, you're exposed to it no matter what, whatever you are. So.
And how about dissemination then, kind of in terms of in a place where it's not available? I'm curious if you have any insights into how the kind of this community coming from so much affluence and having that responsibility, right. How do you see that playing out?
It's difficult again. As you know, a Latin American country or any other third world country around the world, we end up paying for devices 3 times that we pay here in the States. And it's because of all the organization of the system. In regards to the stents, I was interested, for example, on one stent, not Johnson & Johnson stent, but another stent, a very low-tech stent that actually could be manufactured with wires and pliers, that was implemented in China. And it was a fascinating technology that it was very simple, and it was successfully implemented in a country with poor resources. But again, those stories, unfortunately, are getting less and less relevant, because now everything is high technology, and unfortunately, I don't know the answer to that. But I think underdeveloped countries will have a lot of problems keeping up with the pace.
Thanks for your insights.
I couldn't resist asking you a question. Picking up on the early clinical trials, and when you first learned about subacute thrombosis, and that started to be an issue, I was thinking that must have been a difficult time, and I wondered how you responded to that. What did you do when you first learned about the subacute thrombosis cases?
Well, it was very disheartening. As you know, the first one that reported on that was Patrick Cerise, and on the wall stent trials in Europe, they came up with a subacute thrombosis rate of about 25%. So that was terrible. And right after that, I think it was there were a couple of editorials that came out in the New England Journal of Medicine, and then the Wall Street Journal came up with an article on that, and we thought it was pretty much the end of stents. It was a very, very severe blow. What saved the stent program at that time was that the few investigators that were trying our stent and others, balloon expandable stents, around the world, people like Nobuyoshi in Japan, for example, they were started making their own experience and saying, no, this is not true. It's not 1 out of 4 patients is going to have subacute thrombosis, much less. You just have to do it right. And so, that essentially saved us and gave us enough courage to keep going.
Were Patrick Cerise's high subacute a result of practitioner technique, or was it just an outlier study? Or
no, they used self-expanding stents, and self-expanding stents at that time didn't do well in the coronary circulation. But it was just the unfortunate fact that they used the wrong stent in early trials, and the results looked so terrible.
As a follow-up to that question, when you said your motivation was originally to do research for developing the stent, was there ever a time when you thought that maybe you could do more harm than good by developing this device?
You see, by the time we got into clinical research, I had lots of help. I was alone only in the very early phases. But when we got to clinical research, I was just part of a big group. So it was just whatever the group consensus was. And it's always your friends that carry you along. I would have dropped the project a million times. You know, with every complication we had, I would have stopped. But you know, it's always the others that come and tell you, no, but yeah, you had problems, but look at how many people we helped. I mean, how promising it is here or there, and that keeps you going.
In fact, the enthusiasm of Richard Schatz was an important.
Richard is an incurable, enthusiastic person. I mean, he's so optimistic, and yeah, his optimism was a very, very important part of the project.
Hi, my name's Verna Manti, and I've been in the dilatation catheter business for some time. One thing I wanted to ask you about was about the early dilatation catheters and some of the technology that was surrounded the perfusion catheters, where you could leave a balloon up for a long time. Because I remember before stents took hold, one of the things was that, well, dilatation catheters would work if you could hold that balloon up there and let the stuff get pushed aside for a long time. And I didn't know if you had used a lot of perfusion catheters back in those days, but in thinking about it, I was wondering if you might comment about that and maybe the downfall, because obviously I don't think perfusion catheters are used at all anymore, and some of the time that it took to do a procedure, and how stents somehow, you know, obviously are a little bit quicker to use than, say, some of those early dilatation catheters that had perfusion. Because I think that was I thought that was a little bit of importance, because stents make the procedure quicker for doctors.
That's fascinating technology. The perfusion catheter, I always thought that it had a great future. Same with the, you know, the combination of balloon and laser, and the cutting balloon, and the thermal, you know, the balloon, or the cryoplasty and so forth. All those technologies are very interesting technologies. The only problem is that cannot compete with a stent, because a stent is so easy to use and so effective that perhaps if there were no stents, they would have had a chance. But I think in the face of stents, any of those techniques are going to have a real hard time competing.
Great. Any other questions? Anybody else have a final question or so? Well, if not, Dr. Palmaz, thank you very much. Thank you.